Hi guys, we're continuing with life science and cell biology in a nutshell. We're learning about metabolism. This primarily occurs in the mitochondria of both animal and plant cells. So, mm, the electron transferring molecules of the respiratory chain and the enzymes responsible for ATP synthesis are located in and on the inner membrane of the mitochondria called the crista, um, while the space inside, called the matrix, contains enzymes of the TCA tricarboxylic acid cycle. The enzyme systems were primarily responsible for the release and subsequent oxidation of reducing equivalents are thus closely related, so that the reduced coenzymes formed during catabolism or breakdown of molecules, such as the molecules of NADH, hydrogen, and FADH2, are available as substrates for respiration. So, as we mentioned earlier, molecules released energy yielding molecules which come out as a result of catabolism or breakdown of large molecules are used for the process of anabolism or the creation of larger molecules from smaller components in a cycle, so in the cell. The movement of most charged metabolites into the matrix space is mediated by a special carrier protein by special proteins in the crista that catalyze exchange or diffusion, a one-for-one exchange. The oxidative phosphorylation systems of bacteria are similar in principle, but show greater diversity in the composition of their respiratory carriers. The mechanism of ATP synthesis is the following. During the transfer of hydrogen atoms from FMNH2 or FADH2 to oxygen, protons, H plus ions, are pumped across the crista from the inside of the mitochondria to the outside. Thus, respiration generates an electrical potential and, in mitochondria, a small pH gradient across the membrane corresponding to 200 to 300 millivolts. And the chemical energy in the substrate is converted into electrical energy. Attached to the crista is a complex enzyme called ATP synthetase that binds ATP, ADP, and phosphorus ions. It has 9 polypeptide chain subunits of 5 different kinds of these subunits in a cluster, and a unit of at least 3 more membrane proteins composing the attachment point of ADP and P phosphorus. And this complex forms a specific proton pore in the membrane. When ADP and Pi are bound to ATP synthetase, the excess of protons, hydrogen plus, that has formed outside of the mitochondria, the gradient of hydrogen positive, moves back into the mitochondria through the enzyme complex. The energy released is used to convert ADP and Pi to ATP. In this process, electrical energy is converted to chemical energy, so the reverse, and it is the supply of ADP that limits the rate of this process. The precise mechanism by which ATP synthesis complex converts the energy stored in the electrical hydrogen positive gradient to the chemical bond energy in ATP is not well understood. The hydrogen gradient may power other endergonic or energy requiring processes besides ATP synthesis, such as the movement of bacterial cells and the transport of carbon substrates or ions. ATP formation during photosynthesis. Photosynthesis, though this only occurs in plants, obviously, generates ATP by a mechanism that is similar in principle to what we just learned about the metabolism of ATP synthesis, which occurs in animals. So, in principle, they're similar, if not in detail. The organelles responsible are different from mitochondria, but they also form membrane-bounded closed sacs called thylakoids. These are in chloroplasts, often arranged in stacks called grana. Solar energy splits two molecules of water, H2O, into molecular oxygen and four protons and four electrons. So O2 plus 4H plus 4 electrons. This is the source of oxygen evolution, clearly visible as bubbles from underwater plants in bright sunshine. The process involves a chlorophyll molecule that changes its reductive potential from positive 820 millivolts, in which there's a tendency to accept electrons, to about minus 680 millivolts, in which there's a tendency to lose electrons. So the chlorophyll molecule, upon excitation with light and acquisition of electrons. So from accepting electrons to losing electrons, the electrons are subsequently passed along a series of carriers such as plastoquinone and cytochromes B and F and plastocyanin, analogous to the mitochondrial respiratory chain. This process pumps protons across the membrane from the outside of the thylakoid membrane to the inside. Proteins H+ do not move freely across the membrane, although chloride ions Cl- do, creating a pH gradient. An ATP synthetase enzyme similar to that of the mitochondria is present, but on the outside of the thylakoid membrane. Passage of protons H+ through the membrane, through the synthetase enzyme. So from the inside to the outside of membrane, the protons pass, generating ATP. Hence, a gradient of protons H+ across the membrane is the high energy intermediate for forming ATP in plant photosynthesis and in the respiration of all cells capable of passing reducing equivalents such as hydrogen atoms or electrons to electron acceptors. So we're going to continue in the next part. Thank you.